Hello.
Hi.
Okay, just a moment. Let me set everything up. Today we're going to study biology, right?
Yeah.
Okay, what was I teaching last?
We were studying enzymes, and we were doing, I think pH. We finished temperature on enzymes, and then you were supposed to draw the pH graph, I think.
Oh, okay, yeah. All right, so let's begin. Let me check. Just a moment. Yeah, so did I explain everything about the temperature graph?
Yeah, I think so.
Okay, yeah. All right, then let's begin with the pH graph. So, I'm just going to take it a bit slow today because I'm a bit sick. um, all right, so how does the graph look like? So if you plot the rate of reaction on the y-axis and pH on the x-axis, you'll be able to see a sort of a normal curve. So like that. So obviously this is the optimum pH. Okay. Now, yeah, this specific one that I drew, if we have like, let's say, the optimum pH at pH 7, then we are actually talking about amylase. Okay, so this is the graph of amylase, which breaks down starch into glucose. So yeah, they actually work the best at pH 7. Okay, and yeah, the pH right here would be around 4.5, and here about like 9 or something. So yeah, so obviously, as you can see in the graph, as we increase the pH, the activity will start to increase, and then it will reach a maximum at a certain pH, which is called the optimum pH. And then if we increase even further, it will actually, the activity will decrease as well. Yeah, so why is that the case? Okay, I'm going to explain that. Well, yeah, So obviously at low pH, even, both at low pH and high pH, so even right here, the enzyme does not really work. Why? Because at both of these extreme pH, so both acidic and alkaline conditions, the enzyme activity will be distorted. So how? The hydrogen bonds and the ionic bonds that actually hold the 3D structure of the enzyme, I mean the protein, that will be disrupted. So let me just say something like this. Enzyme, which is a protein, are disrupted. So what happens if the shape is distorted? The active site is changed, and even at the extreme level, the enzymes can get denatured. So yeah. That's the explanation. So at extreme pH levels, the enzyme is completely denatured and the rate of reaction is very low. Okay.
Okay. The enzyme will only be denatured when it is more alkaline, like after 7, or does it happen if it's too acidic as well?
Yeah, it happens if it's too acidic as well. So for example, in the case of amylase, as you can see, it will only work properly at pH 7. It will work the best at this pH. It's okay if you reach a pH of, let's say, like, I don't know, 5 or 6. Let's say 5.5, or even this is okay, which is like, I don't know, 7.5. But as you can see, the activity completely stops at 4.5, which is not even that acidic, because yeah, something that is extremely acidic is probably like around 3 or 2 or even 1. And 9, so even for amylase, even a weak acid or a weak alkali will completely denature it. So it's very sensitive to both the pHs, I would say. It is only safe to work around this range, 5.5 to 7.5.
Yeah. And that with the temperature, it's the same, right? If it's too less, like the temperature is too less, it will get denatured, and if the temperature is too more, it will also get denatured.
Well, in the case of temperature, yeah, it is actually the same. But in the case of temperature, um, obviously we mentioned that at higher temperatures, the enzyme starts to denature, and when it's at a very low temperature, then I wouldn't say it is denatured, but I would say it's too inactive. It doesn't have enough energy to work. Okay, so yeah.
I copied it.
All right. Yeah. Now, one thing to note is that the optimum pH will differ for every enzyme. Well, every type of enzyme. Okay, and I'll give you some examples. Like pepsin, it works best under acidic conditions inside our stomach. And there's another, I cannot recall their names, but you know, the intestinal enzymes, they work best under alkaline conditions. Okay, that's it.
Come down.
All right. I think we're done with enzymes, right? Let me check with the next chapter in your syllabus. Okay, I think you have plant nutrition and animal nutrition, right?
Yeah.
Okay. So let's start with plant nutrition today. Okay, let me see, what do you have in plant nutrition? Photosynthesis, leaf structure, mineral nutrition. Others under photosynthesis, the fundamental process. Necessity for chlorophyll, light, and carbon dioxide using appropriate control state equation. Investigate and state the effect of varying light intensity, carbon dioxide. Okay. Understand the concept of limiting factors. Describe the intake of carbon dioxide and water by plants. Understand the chlorophyll traps light energy. And know why most forms of life are completely dependent on photosynthesis and endogenous cuticles, cellular and tissue structure for dicotyledonous leaf. Okay, great. Let's do that. Okay. But I think let's start with the leaf structure. Obviously, if we start with the external leaf structure, which is external leaf structure means, you know, how does the leaf look, you know, from the outside. So obviously we have this leaf blade or lamina. This is, you know, it's quite thin and obviously has a very large surface area to volume ratio. This is very much needed so that, you know, sunlight can be, you know, a lot of sunlight can be easily absorbed for photosynthesis. and also because it needs to diffuse oxygen, I'm sorry, oxygen, and also take in carbon dioxide and obviously sunlight. Okay. Yeah, so I'm just gonna write that down. All right. So that is about leaf blade. Oh, by the way, a leaf also has a stalk. So like somewhere here. Yeah, so the leaf stalk, I don't know if you need to know the other name. I'm just going to mention it anyway. Also known as petiole. What does it do? It holds the leaf away from the stem so that the leaf can get more sunlight, right? All right. Now this mid thing is called a midrib or a vein network. Sorry, this is a vein network. I'm sorry. This is a midrib. What does it do? Why is it there? It carries food substances away from the leaves, okay, and the water and mineral salts to the leaves. So mineral and water salts, they enter through this stalk and they go all the way like that. And then the food, which is made by the leaves, are actually going backwards like that, okay? So if you want me to label all of these, so this right here is the leaf stalk. This one is the midrib. This one is a lamina. This one is the tip. What else? These ones are veins. Okay.
Yeah. One done.
Okay, great. And I can move on to the next part, which is the internal leaf structure. Okay, so let's do that. This is quite a complex one, but still let's try to draw. Yeah. So what does the internal leaf structure consist of? It actually has multiple layers. So for example, the first one, so this layer is called the upper epidermis. I'm going to label it later on. And then we have these longitudinal cells. Okay. I don't know, you have to know these in details. Have you ever heard about palisade mesophyll?
Yeah. I think we have to label them and write.
Okay. Okay. So these are the cells that actually have chloroplast. So the dotted things that I'm drawing, those are actually chloroplasts. Okay. And then we have this spongy mesophyll layer, which is not longitudinal and not so packed. So it's like this, kind of like circular, not really circular, but irregular. Okay. Now the space right here that I've actually kept for, these are for the vascular bundles and xylem and everything. So a stem that goes through this. Okay. And right here, the last part is the lower epidermis. So this one is simple. Now, another thing is that the spongy mesophyll cells, they also have chloroplasts. So okay. All right, let's first label these for now. Then I'm going to include the vascular bundle. Um, so this right here is the upper epidermis. And on top of the upper epidermis is the cuticle, which is like this waxy layer. Yeah, so I already mentioned the dotted things are chloroplasts. And yeah, this layer right here, this layer is actually called palisade mesophyll. Okay, like I said, these ones are lower epidermis. And the lower epidermis does not have a cuticle. যেটা তুমি maybe like notice cursor if you have touched a leaf, the underside is really rough. So because there's no cuticle layer. This part right here, this is spongy mesophyll because this has more gaps. It's not so tightly packed. All right. Now, one thing to note, I'm just going to color this in a different way. This right here, these two are called guard cells. We're going to study this in details. So yeah. Okay. And this opening right here, which is this one, this is stoma. Yeah, plural is stomata. I think you've heard this before.
Yeah.
Okay. And now let's draw our xylem, phloem, and the entire vascular bundle. But can you give me a minute? I'm just going to come back. Okay.
Yeah, sure.
Okay, I'm back. Yeah, so let's draw our vascular bundle. Right. So it should be actually, yeah, let's do it in a different color. I'm gonna do it in yellow. And then, you'll see like, just a moment. Mm. You'll see that in the middle, there is like, a bundle of very small tubes like that. Yeah, so this right here, let's see. This entire thing right here, this is the vascular bundle. Right here, somewhere we have xylem, and we also have like color them. We also have phloem, which you'll learn in details, and then you'll be able to identify them easily. Okay. Are you done drawing them?
Not yet. I'm drawing it.
Okay.
I'm done.
Okay. I'm trying to find some, like 3D drawings so that it's easier for you to see in different angles. Yeah, I got one to show. Okay. I've collected two 3D drawings so that you can understand it better. So I'm just going to show it here. I think if you look at this picture, it will give you a better idea of everything in a 3D structure. So we have this cuticle on top, and then we have upper epidermis, then all the mesophyll cells. They did not really divide it into palisade and spongy, but I'm sure you can identify. And then on the low, there's lower epidermis, and then we have stoma and the guard cells. Yeah, in the middle, there's a vascular bundle, and inside the vascular bundle, you can clearly see um, the two types, so the xylem and the phloem, right? And yeah, that's it. Another picture right here. Again, you can see the cuticle on top. And yeah, then we have this upper epidermis, and then palisade mesophyll and spongy. Then we have the guard cells, the stoma, the lower epidermis. Also, they showed a cuticle on the lower epidermis, which is okay. And then the bundle of, there's this bundle of vascular tissue. So you can see that there are multiple pipes.
Does the lower epidermis always have cuticles, or it's just in some certain leaves?
I think in some certain leaves, because usually leaves have cuticle on the upper epidermis, not usually on the lower one. But yeah, it could be the case. I mean, there are always exceptions.
Yeah.
Yeah. Okay, that's it. Now we can learn everything in details, I guess. I hope now you can label everything clearly, because yeah, there are questions where you actually have to label them. Yeah. All right. Okay, now let's talk about them in details. Let's talk about the upper epidermis first. So what did it look like, the upper epidermis? Obviously, it was a single layer of, closely packed cells, right? Covered by a layer of waxy cuticle. Okay.